CRISPR-Cas9, reshaping our food landscape. Imagine a future where bananas never brown before you finish them, wheat fields resist drought and disease without chemical pesticides, and livestock grow with better health and lower environmental impact. Now imagine that all of these changes aren't the result of decades of slow breeding, but of precise, targeted edits to DNA edits, so accurate that we can change a single letter in the genetic code. This is the promise of CRISPR-Cas9, a revolutionary genetic engineering tool discovered just over a decade ago. Initially hailed for its potential in human medicine, CRISPR is now reshaping our food systems, from crops and livestock to the microbes that ferment our food. But with great power comes great responsibility. The ethical, ecological, and economic implications are as profound as the science itself. The science of CRISPR-Cas9. To understand its impact on food, we must first understand what CRISPR-Cas9 is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's part of a bacterial immune system that evolved to fight viruses. How it works. Cas9 is an enzyme that acts like molecular scissors, cutting DNA at a location guided by a short RNA sequence. Scientists can design this guide, RNA, to match any sequence in the genome. Once Cas9 makes the cut, the cell's natural repair mechanisms either disable the gene or insert a new piece of DNA. Why it's revolutionary? Precision. Unlike older genetic engineering methods, CRISPR can target specific genes without introducing random changes. Efficiency. Edits can be made quickly, sometimes in a single generation. Versatility works in plants, animals, and microorganisms. The result? A genetic word processor that can rewrite life's code. Applications in crops. CRISPR is already transforming agriculture at multiple levels. 1. Disease resistance. Rice CRISPR has been used to knock out susceptibility genes that make rice vulnerable to bacterial blight. Cacao researchers are editing cacao trees to resist Phytophthora fungus, potentially saving chocolate production. 2. Climate resilience. Drought-tolerant varieties of wheat and maize are being developed by editing genes, controlling stomatal opening and water efficiency. Altering flowering time in tomatoes genes to adapt to shorter growing seasons. 3. Nutritional enhancement. Japan has already commercialized a CRISPR-edited tomato rich in gamma-aminobutyric acid, linked to reduced blood pressure. Editing biofortified cassava to reduce cyanogenic compounds and increase micronutrient content. 4. Reduced waste. In the US, a CRISPR-edited mushroom resists browning, extending shelf life without chemical preservatives. Applications in livestock. CRISPR is not limited to plants, it's influencing the way we raise animals. Disease resistance. Editing the CD163 gene in pigs makes them resistant to porcine reproductive and respiratory syndrome, or PRRS, a major economic threat. Research aims to create resistance in chickens to avian influenza. Welfare improvements. Hornless cattle. Editing horn genes to prevent the need for painful dehorning procedures. Heat-tolerant livestock. Introducing traits from heat-adapted breeds into high-yield dairy cattle. Productivity and sustainability. CRISPR can reduce methane emissions in ruminants by altering gut microbiota genes, potentially reducing agriculture's carbon footprint. Microbial and food processing innovations. CRISPR's reach extends into the invisible world of microbes that make cheese, beer, yogurt, and other fermented products. CRISPR in starter cultures. Engineering lactic acid bacteria to resist phages, viruses that can ruin fermentation batches. Flavor optimization. Editing yeast to produce new aroma compounds or reduce off flavors in beer and wine. Food safety. Engineering bacteria to detect and neutralize contaminants like listeria in food processing plants. Ethical and social implications. 1. Regulatory ambiguity. In the US, some CRISPR-edited crops, like non-browning mushrooms, bypass GMO regulations because they contain no foreign DNA. The EU, however, currently regulates CRISPR-edited plants as GMOs, sparking debates about innovation versus caution. 2. Consumer acceptance. Surveys show mixed public opinion. Higher acceptance when the edits improve nutrition or reduce pesticide use. Lower acceptance when changes feel unnatural. 3. Intellectual property and equity. CRISPR patents are controlled by a handful of institutions, raising concerns about monopolization. Small farmers in the global south risk being left out unless technology is shared affordably. 4. Ecological risks. Gene edits could unintentionally spread to wild relatives, potentially disrupting ecosystems. Over-reliance on single traits could reduce biodiversity, making crops vulnerable to new diseases. Case studies. Japan's GABA tomato. First, commercially sold CRISPR-edited food, marketed for its health benefits. No special labeling required. US non-browning mushroom. Passed USDA approval because it had no transgenes, setting a precedent for minimal regulation. African cassava project. Aims to use CRISPR to combat cassava brown streak disease, a major threat to food security. These cases illustrate both the promise and complexity of deploying CRISPR in real food systems. The future outlook. Looking ahead, CRISPR could enable polygenic editing, simultaneous changes in multiple genes to create complex traits like drought plus pest resistance. De novo domestication. Turning wild plants into crops in a few generations by targeting domestication genes. Smart breeding systems. Integrating CRISPR with AI to predict the best gene targets for specific climates. On-farm CRISPR toolkits, allowing farmers to make minor genetic adjustments in real time. Though this raises major regulatory questions. Balancing promise with responsibility. CRISPR-Cas9 offers us the ability to reshape the food landscape with unprecedented precision to create crops that thrive without harming the planet, livestock that live healthier lives, and food systems that nourish billions more people. But science doesn't exist in a vacuum. The future of CRISPR in food will be determined not just by what is possible in the lab, but by how we navigate ethics, regulation, and public trust. If we get it right, CRISPR could be remembered as the green revolution of the 21st century, one built on code, not chemicals. If you found this video interesting, imagine what's coming next. Subscribe for more science-driven insights into the future of food, health, and technology, because your next meal might just be your most personalized one yet.